Today I'm describing the role of enzymes in metabolic reactions. All cells perform certain basic reactions, such as the buildup and breakdown of carbohydrates, lipids, proteins, and nucleic acids. Common reactions include specific chemical changes that must occur in a particular sequence rapidly enough to sustain life. Enzymes help speed up the rate of these chemical reactions. Metabolic reactions require energy to proceed. Kind of like, for example, how heat can be applied to speed up chemical reactions by increasing the rate at which molecules move, which increases chances of collision and interactions between electrons that form new chemical bonds. Cell's temperature does not get hot enough to start the chemical reaction, and that's where enzymes come into place to help this happen. Globular proteins are the most common enzyme, and they catalyze or cause the process to begin by lowering the energy required to start the reaction. Did you know that enzymes can speed up the metabolic reactions by a factor of 1 million or more? Catalysis is the acceleration of chemical reactions in which the enzymes in the catalyst Enzymes are only needed in small amounts because they work and are not consumed, so they can function repeatedly. So basically the enzyme can keep being used over and over again. Just a few enzymes have a major effect, and each enzyme is specific to a particular molecule type, which is its substrate. In this example here, the substrate of the enzyme called the catalase, which is in the peroxisomes of liver and kidney, is hydrogen peroxide, which is a toxic byproduct. The catalase enzyme decomposes hydrogen peroxide into water and oxygen, as you can see in the example. Without the catalase, hydrogen peroxide will accumulate and damage. Many enzymes are derived from the substrate name, like lipid, which is splitting enzyme into lipase. The role of an enzyme in an enzyme catalyzed reaction is part of the active site temporarily combines with the parts of the substrate molecule, which forms an enzyme substrate complex. When this happens, the enzyme is released in its original conformation and can bind to another substrate molecule. Many enzymes catalyzed reactions can be reversed. Sometimes the same enzyme catalyzes in both directions. You can look here and you can see how the substrate and the enzyme combine, and then it gets to the substrate complex and the product, and then the enzyme comes out at the end unchanged. The rate of an enzyme catalyzed reaction depends partly on the number of enzymes and substrate molecules in the cell. The more enzyme substrates equal a faster reaction. If exposed to heat, radiation, and certain types of chemicals a lot, it can be like a devaluation of the actual enzyme. So anything over 45 Celsius, which is 113 Fahrenheit, is too hot. Thank you.